I'll go ahead and do introductions. My name is Bill Walton. I'm at the Auburn University Shellfish Lab, and pleased to kick off this semester's seminar series, hosting Dr. Andrea Tarnecki. Some of you may know her, but most recently, she is a staff scientist at the Mote Marine Lab in Sarasota, Florida, where she also did an extended postdoc there as well. Before she started her postdoc, she did do her graduate work at Auburn University. I was lucky enough to serve on her committee, and she was lucky enough to spend her time down here, most of her time for her graduate work down here at the coast. And so she was actually at Dauphin Island Sea Lab. So some of you, I think it was Andrea Larson at that point, right?
Yes, that's right.
Andrea, actually, some of you may know her husband who worked here at the Sea Lab, Joe Tarnecki. The two of them moved to warmer clime in Sarasota, along with their 3 dogs, Roddy, Crouton, and Bakos. And I know, because I'm friends with Andrea on Facebook, I know they spend a lot of time fishing. Today, she is going to talk about fish, but instead of how to catch them, she's going to talk a little bit about the microbiota in fish and thinking a little bit about healthy bacteria, or maybe the ones that pose a problem. I know that Andrea cares quite a bit about bacteria, because whenever I get an email from her, her signature line, let's see, what is it, Andrea?
If you don't like bacteria, you're on the wrong planet.
You're on the wrong planet, Craig Venter. So Andrea, I will hand that over to you, and appreciate you taking the time to visit with us, even if it's remotely.
Yeah, thank you, Bill. It's great to kind of be back, I guess. Yeah, I'm not going to lecture you on fishing because I am not the best person to do that, but my husband would be. Let me figure out which screen to share. It's not that one. I guess this one. There we go. Okay, so thanks for having me for your first seminar of the semester. Today, I'm going to talk kind of a general overview of my research at Mote Marine Laboratory. So I'll be talking about unraveling the mysteries of marine microbiota. Just technical difficulties. Okay, for those of you unfamiliar with Mote Marine Laboratory and Aquarium, we are located in Sarasota, Florida, which is Central Florida on the west coast there on the Gulf of Mexico. The laboratory currently has over 200 employees and 30 plus PhD scientists. I think we bring in new postdocs on occasion, so that number changes. But those employees and PhDs run 20 plus diverse research programs, ranging in everything from chemistry and oceanography to biotechnology, and then all the way up to your marine mammal realm. Specifically, my position is microbiology division lead within the marine immunology program. My laboratory is not located on Mote's main campus, which hosts most of the research programs as well as the aquarium, but actually at Mote's Aquaculture Research Park, which we lovingly call MAP. This is an inland facility. You can see an aerial view of it there on the bottom right. The facility is 200 acres, which currently about 15 acres are being used. The farm has all aspects of fish rearing associated with it. So we do have brood stock in the systems that we use for spawning to create animals for research and stock enhancement. The species that we have currently are red drum, common snook, and the Almaco jack. And on occasion, there are also Florida pompano. Also on the facility, we have an aquaponic system, which rears fish together with sea vegetables, more specifically sea purslane. So there's a lot of different research that occurs at the park, ranging from stock enhancement to reproduction and optimizing aquaculture protocols. And since my lab, although I cover a lot of microbiology, I generally focus on the aquaculture world, I've kind of set up out there, and they're stuck with me at that facility. So now that you know a little bit about where I'm located, we can jump right into the research in my program. So the main question here, what is the microbiota? Well, the microbiota is defined as the complex community of microorganisms that are associated with a particular habitat. So fish skin and gastrointestinal tract have a microbiota or microbial community. All the surfaces in the aquatic environment do, as well as all the surfaces on every living organism. These communities are extremely complex and are made up of many species of bacteria, as well as eukaryotic microbes and archaea as well. And my focus is mainly on the bacterial component of these microbiota communities. The term microbiota more specifically refers to the species that are present and their relative abundance within that community. The microbiota is an important area of research because we know that is absolutely vital to aquatic animal health. And it's especially important for these aquatic organisms because they're in the water. The water has pathogens living in it. So these organisms are in direct contact with potential pathogens at all times. But a healthy microbiota, which is one that is composed primarily of commensal and mutualistic bacteria, has a number of roles that help to protect that organism from pathogens and also to improve overall health. Most of what's known about the role of the microbiota are on the nutritional effects. So the healthy microbiota is important from developmental standpoint to develop a properly functioning gastrointestinal tract. These microbes also produce digestive enzymes and supplement the nutrition of the host. The bacteria also have a number of immunological effects. They can detoxify pollutants and xenobiotics. They stimulate immune function, and they basically provide an extension of the immune system by establishing on the different surfaces of the aquatic animal and outcompeting the pathogens that are present. So they really do act as an extension of the immune system to prevent infection. More recently, there's been work into the neurological effects of a healthy microbiota, and some of those effects that are known include reduction of anxiety and heightening predator awareness. Depicted here at the top is what we would call a healthy microbiota structure. This community is dominated primarily by probiotics, which are beneficial bacteria that provide any of the benefits that I mentioned in the previous slide, as well as commensal microbes, which again do not cause any harm to the host. So between the probiotics and the commensals, these help to outcompete the pathogens, which are depicted here by this little red cell with the many flagella. But many things can happen during the course of an organism's life that may change the structure of that microbiota. For example, the animal may come into contact with a pollutant, it may become immunosuppressed, and it can alter this healthy microbiota structure to one that is more dominated by pathogens and has a much lower abundance of probiotics and commensal microbes. This is kind of a degree of change and an alteration from that healthy microbiota structure to one that's dominated by pathogens is known as dysbiosis. Dysbiosis has been associated with disease, and the greater the dysbiosis or change from normal, the greater the disease severity, to the point where the host organism can experience increased mortality rates, an accumulation of toxins, an immune stress response, which may be immunosuppression, or it could be the opposite, which would overstimulate the immune system and cause excessive inflammation. Despite what we know about the microbiota, there's still a lot that we really don't know, and that brings me to the purpose of my research. My research can broadly be broken down into two long-term research goals. The first is to establish the links between microbiota structure and aquatic animal health. So in this goal, I'm looking to decipher what a healthy microbiota looks like versus a dysbiotic one, and what factors lead to those shifts in microbial community structure, as well as what effects those have on host health. The second goal is to develop protocols to maintain or restore host homeostasis. So if we do have an aquatic organism that's experiencing dysbiosis, are there ways that we can alter that microbial community to get it back to a healthy structure? Or if not, are there things that we can give to the animal in order to mitigate the negative impacts of dysbiosis? So in today's presentation, I'm going to be going over 5 projects that are related to these 2 long-term research goals. The first is investigating dispersed oil and the skin microbiota of red snapper. And the second is looking at alternative fish meal sources and their effects on fish production and the gut microbiota of different fish species. And both of these studies kind of fall into that first goal of establishing the links between microbiota and animal health. The third project I'll talk about is the use of probiotics or beneficial live bacteria in larval fish rearing. Then I'll talk about some research that I've instigated in the last year or so, looking for new bacterial species in the aquatic environment. And finally, bioprospecting for beneficial compounds within naturally occurring environmental bacteria. So let's jump right into the first study. The objective of the first study I want to talk about was to quantify the health and microbial effects of crude oil and dispersant exposure on the skin and immune function of the red snapper. So many of you probably remember after the Deepwater Horizon oil spill, there was a large increase in reports of red snapper with these external lesions on their bodies. So the red snapper kind of became the poster child for the potential negative effects of a crude oil spill. So the goal of this was to see if we could manipulate those lesions and get them to show up in an experimental form. The red snapper in our study were exposed to the chemically enhanced water accommodated fraction, or CEWAF, which contained extracts from crude oil and dispersant. The exposure was set up according to the grid on the bottom right there with 6 different treatments, 2 of which kind of mimicked controls in which the fish were in clean seawater for 28 days. Half of those received a bacterial challenge after the first 7 days and then were allowed to recover for 3 weeks. The second 2 exposures were set up as recovery, so we could see if there are changes in the microbiota and immune status, if those return to normal, if the fish are replaced into clean seawater. And again, half of those fish also received a challenge of bacteria. And finally, we had 2 exposure treatments where the fish were in the CEWAF for the entire 4 months. So it was a 28-day experiment. The goal for the concentration of the crude oil and dispersant was 1 part per million total petroleum hydrocarbons. And the challenged fish were challenged with the fish pathogen Vibrio anguillarum. Looking at the accumulation of polycyclic aromatic hydrocarbons, which are one of the main constituents of crude oil and also known to be toxic, we did get an accumulation of the PAHs in the livers of our fish. And you can see that the fish that were in the CEWAF for the entire 28 days, there, the 2 bars on the right, had the highest concentration of PAH accumulation. So these fish were accumulating polycyclic aromatic hydrocarbons over time as they were exposed to the dispersed oil. To look at immune function, we measured immunoglobulin M expression using quantitative PCR. IgM is the main antibody that's produced in fish. And these were measured again in the spleen. So what you see from this graph is that the IgM expression was significantly higher in fish that were given the bacterial challenge. So those would be our bacteria controls, our recovery bacterial challenged, and the exposure bacterial challenged fish. However, you'll see that the fish that were in CEWAF for the entire 28 days had lower IgM expression than the fish that were in seawater following bacterial challenge. So what this suggests is that continued exposure to dispersed oil suppresses the immunoglobulin M expression in the spleen of chronically exposed fish. So my major role in this project was to look at the effects of the CE WAF on the skin microbiota of the red snapper. This is an ordination plot where each of the dots represents the microbiota structure of an individual fish. So each dot is a fish. And as the fish cluster together, that tells you that their microbiotas are more similar in comparison to other fish. So you can kind of see here that the fish did tend to group by our treatments, with the control and bacteria treatments at the bottom right, the recovery treatments at the top right, and the CE WAF chronic exposures on the left. So some of the bacterial groups that lead to these differences kind of shed some light on the potential effects of this dysbiosis or shift in the microbiota community due to CE WAF exposure. The bacterial groups that were dominant in the seawater and bacterial treatments there on the bottom right included normal members of the marine fish microbiota, and many of them are reported to have potential probiotic functions, including acting as immunostimulants, producing vitamin B12, and also producing natural antibiotics that can help resist disease. In addition, a number of these taxa have been demonstrated to reduce the abundance of the Vibrio bacteria that include not only Vibrio anguillarum, which is the bacterium we used for our challenge experiments, but also a number of other opportunistic fish pathogens. So these are generally what we would consider probiotic or commensal microbes. If we look at the recovery fish, what we see are that these groups that are dominant in these fish are also normal members of the marine fish microbiota, but the literature reports them as potential opportunistic marine fish pathogens. What that means is that these bacteria are naturally present in the skin microbiota of fish when they're healthy, but during times of immunosuppression or other incidences that alter host physiology, these marine bacteria can become pathogenic and cause disease. So we're getting this shift from probiotic and commensals to opportunists, and also highlighted here in the bright green from the bacterial challenge recovery was a fish that actually developed an external lesion during the course of the study. Looking at the microbes that become dominant in the chronically exposed fish, we see that there's a shift from opportunists to different opportunists, and also groups that are known to be primary fish pathogens, including the streptococcus. So again, we're getting this dysbiotic shift with chronic oil exposure that leads to the selection for potential fish pathogens. And again, here highlighted in green is another individual from the CE WAF only exposure that developed an external lesion. An interesting pattern that came out of the microbiota analysis was that the potential hydrocarbon degraders found in the skin microbiota of exposed red snapper reflected patterns that are seen during oil plumes that happen in the natural environment or in mesocosms. If we start on the right with our chronically exposed fish, you see the increased abundance of the alcanovorax bacteria, which are these black bars. And the alcanovorax are known to be primary hydrocarbon degraders. They are often seen at the beginning of an oil plume. And we hypothesize that because the CE WAF is still being added to, and the fish are being exposed to it at this time, that those primary compounds are still available for the alcanovorax to degrade. Looking at the recovery fish, we get a spike in the Pseudoalteromonas and the Coelia bacteria. These are recognized in the literature as secondary hydrocarbon degraders. So after those primary constituents are broken down, the Pseudoalteromonas and Coelia come in and continue to break down the remaining hydrocarbons in the plume. So the skin microbiota of red snapper is reflecting what would happen if you were measuring a microbiota composition in the water during a bloom, which means that the skin microbiota structure can potentially point you to previous exposures to pollutants. I mentioned the 2 fish that developed lesions during the course of the study. This is on the bottom right is a picture of the most dramatic lesion that we had. So we only had 2 individuals in the microbiota sampling to compare between lesioned and non-lesioned fish, but it did provide a really interesting potential pattern. In the lesioned fish, you see a much higher abundance of Vibrio bacteria than in the non-lesioned fish. And this is an interesting pattern because not only one of these lesioned fish was exposed to the Vibrio anguillarum pathogen. The other one came from the CEWAF only exposure, which suggests that we may be seeing a different Vibrio spiking up here in these sequences. So in summary, the dispersed oil did suppress the immune response, which was indicated by the reduced expression of IgM in the chronically exposed fish with a bacterial challenge as compared to the recovery and control fish that were also exposed to the bacterial pathogen. The dispersed oil also induced dysbiosis in the skin microbiota in that it selected for opportunistic and primary fish pathogens. The bacterial taxa that were identified as hydrocarbon degraders may act as biomarkers of environmental health, as the microbiota dysbiosis reflected that of oil plume degradation, seeing those primary hydrocarbon degraders shift to the secondary degraders over time. And finally, some CEWAF exposed fish did develop external lesions. These 2 individuals had a much higher abundance of Vibrio sequences than non-lesioned fish. But again, this was not a response to the pathogen challenge, as only one of those fish was actually exposed to Vibrio anguillarum. Therefore, the hypothesis is not that the Vibrio anguillarum caused the lesion necessarily, but maybe the dispersed oil led to immunosuppression that increased susceptibility of these fish to the pathogens. All right, so moving on to the second study. The objective of this study was to characterize the effects of aquaculture feeds on the gut microbiota of fish. So kind of to make a long story short, the goal of this study was to improve the economic viability of the mullet fishery. Mullet is a really large fishery here. It's historical, it has a large economic base, but increasing restrictions on the mullet fishery has limited their abilities to expand their businesses. And the valuable part of the mullet fishery is the roe or the fish eggs and not so much the fillets. So keeping the fillets is not cost effective for the fishermen. So the idea behind this study was to find another outlet that the fishermen could provide their fillets to in order to make more money off of the whole fish instead of just the roe. We tested 3 feeds. One was a standard commercially available feed that changed depending on the fish species we were testing. And then we generated 2 fish-based feeds, one being menhaden and one mullet-based. And the menhaden and mullet replaced the fish meal and oil component of the standard feeds, and then all of the other nutritional components were as similar as possible to the standard commercially available diet. These 3 feeds were tested in a freshwater species, the Siberian sturgeon, as well as a marine species, the red drum. So these results show the effect of the mullet feed on production of Siberian sturgeon. The left bar graph indicates the specific growth rate, which is the percent weight change per day. And you can see that all feeds performed approximately the same. Menhaden did have a faster growth rate than the standard feed, with the mullet kind of in between. If we look at the right side, this indicates the feed conversion ratio, which is how much food it takes to produce a certain biomass of fish. So a lower feed conversion ratio means that the fish is more efficiently processing food into body weight. So it takes less food to produce more fish. There's a trend in the feed conversion ratio for the mullet-based feed to be lower than the standard diet, but this was not significant. Looking at the gut microbiota analysis, this is a stacked bar graph with the relative abundance of the most dominant bacterial groups. In all species, the dominant genus was Cetobacterium, which is indicated here by this purple bar. And you can see that the mullet feed had much higher abundances of Cetobacterium than either the standard or the Menhaden-based diets. Cetobacterium is a really interesting microbe that people are pursuing as a potential probiotic because it is known to produce a high amount of vitamin B12 that can be directly used by the fish. So the mullet feed seemed to have an increase in this potentially probiotic microbe. And that was in the sturgeon. So now if we move on to the red drum, which was our marine fish species, the red drum grew significantly faster on the mullet-based feed than they did on either the standard or the Menhaden diet, shown here on the left. And on the right, the feed conversion ratio was significantly lower for the mullet-based feed than the standard feed, meaning that it took less of the mullet feed to produce more fish. Interestingly enough, the feed did not alter the gut microbiota in a dramatic way. There are some subtle changes, but nothing that would obviously account for those differences in growth. This study demonstrated that mullet can be successfully incorporated into fish feeds and that those feeds can be used in a variety of fish species. However, the mullet-based feed impacts do seem to be fish species specific. There were no differences in growth rate or feed conversion ratio in the Siberian sturgeon, despite an increased abundance of vitamin-producing bacteria. However, we did have that trend towards the decreased feed conversion ratio in the mullet-based feed, so it is possible if that project was extended that may become a significant difference. For the red drum, they did grow significantly better and they were more efficient at processing the nutrients in the feed into biomass, despite no change in the gut microbiota. However, the mullet-based feeds do provide an avenue that can potentially improve the economic viability and sustainability of the mullet fishery by providing the fishermen another outlet for the fillet products. Okay, study 3 is kind of my bread and butter. This was the original topic that I was really interested in when I started my postdoc way back in the day, and that's the use of probiotics in larval fish rearing. The objective for the study was to use microbial management to increase aquaculture production. So if that microbiota in your aquaculture system becomes unbalanced and dysbiotic, it can affect the production of aquatic organisms, particularly larvae, because they tend to be more sensitive to opportunistic pathogens in the environment. So if you can keep that microbial community balanced, it should improve the health of the animals in the system. The management strategy we used for this study was probiotics, which again are live bacterial supplements that can be added directly to the system, or in some cases they can be incorporated into the feeds. The species used for this study was the common snook. Mote has a really strong stock enhancement program where snook are reared and grown up to juvenile stage, tagged, and then released into the environment. So they are very sensitive, however, to environmental changes. So that's why we chose these larvae to work with. And the probiotic used is a mixed species bacillus probiotic that was given to us by our partners here in Sarasota. The experimental design looked somewhat like this. There were 3 separate recirculating systems with 6 replicate tanks each. The first 6 tanks were our controls where no probiotic was added at all. And these fish were fed control rotifers, so rotifers just grown in batch culture and given directly to the tanks. The second treatment received probiotics in the water only, so the probiotics were mixed and dumped directly into the replicate tanks, and these were fed control rotifers also. The third treatment was probiotics in the water and the live feed. These fish received both the water supplemented bacteria as well as rotifers that were grown in probiotics. So they kind of got a potential double dose of the probiotics there. This trial was actually ran twice, but for the ease of presentation, I'm just going to show you 1 year's results because they are actually incredibly similar between the two trials. On the left is total survival of our shrimp larvae after 30 days of receiving probiotic supplementation. The survival in both probiotics treatments was significantly higher than that of fish that received no probiotic. And in fact, this survival was about 2.5 times higher, which equated to 2,500 additional larvae surviving to 30 days in each system. Then we were actually donating these larvae to a local high school for aquaculture education. And so the larvae were bagged up and transported to the school. And we used that as an opportunity to look at the effects of probiotics on transport stress and transport survival. So the fish from each treatment were bagged into plastic bags, transported to the local high school, and then put into new systems. And the survival was monitored immediately after transport and 7 days later. Immediately after transport, survival of the probiotic treated larvae was 10% higher than control larvae. And a week later, that survival was 20% higher. So there was some sort of lasting physiological effect of the probiotic on these individuals. Looking into the microbiota structure, I investigated the structure in both the larvae and their surrounding water at the end of those 30 days. So this is the 30-day mark, and the structure points to some potential benefits of the probiotic. First thing I want to point out is that the purple bars here are the bacillus sequences. These would include the probiotic, and they're found more in the water column than they are in the fish microbiota. So although they are establishing in the fish, it's at very low relative abundance as compared to what's seen in the water. The next interesting thing that popped up, these are Vibrio sequences, and I've mentioned Vibrio a number of times because they have numerous opportunistic pathogens within that genus. What we saw in the fish microbiota was that Vibrio abundances actually increased when probiotics were given. That's seen here on the left in comparison to the control. However, in the water microbiota, the Vibrio sequences decreased with supplementation of the probiotic. That led me to question, what is the effect of the probiotic on specific Vibrio species? So one summer, I had an NSF REU that went around MAP and collected as many Vibrios as he could, sampling from various system waters and biofilters at the park. Also the opportunity to sample fish eggs, red drum and common snook skin, and also Almaco jack larvae, just based on what we were growing during that summer. The Vibrio were collected on TCBS, which is this green agar shown here at the bottom. And the antagonism assays were performed by spotting the bacillus onto plates in triplicate. So on kind of the top right here in this middle picture is one of the bacillus strains, and the bottom kind of left is the other bacillus strain. So they were allowed to grow on the agar surface for a few days. And after that, they were UV sterilized, and the Vibrio isolate of interest was mixed into a soft agar layer and poured over the top of that plate. The idea behind this type of bioassay is that if the Bacillus strains produce a compound that inhibits the growth of the Vibrio, you'll get this nice, pretty clear zone around the Bacillus colony that's known as the zone of inhibition. And that indicates that there's a compound that's diffused into the agar that's produced by the probiotic bacillus that inhibits the growth of the Vibrio. If no substance is produced, you just get this nice lawn of Vibrio growth over the top of your plate as seen on the right. So in looking at the strains that were inhibited, there were no Vibrio strains from the system water or the Almaco Jack eggs that were inhibited by the probiotic. However, 20% of Vibrio strains isolated from red drum skin were inhibited, including one potential pathogenic species, which is Vibrio coriolyticus. 31% of strains from common snook skin were inhibited, including Photobacterium damselae, which also grows on TCBS agar and is a potential fish pathogen. And 36% of strains isolated from the larvae were inhibited, including Vibrio alginolyticus, Vibrio coriolyticus, and Vibrio rotiferianus. So a number of potential pathogenic Vibrios were inhibited by our probiotic strains. So these two studies kind of crammed into one demonstrate that this mixed bacillus probiotic increases survival and transport resistance in common snook larvae. The Vibrio abundances from the microbiota analysis indicated that they decreased in the water column with probiotic supplementation. The hypothesis here is that there's an indirect competition that's occurring in the water because we were not able to get any inhibition of the Vibrios that were collected from various water columns at MAP. So we think that maybe in the water, we're getting competitive exclusion of the Vibrios from the bacillus establishing, particularly in the biofilms on the sides of the water of the tanks, and inhibiting the ability of Vibrios to establish. On the other side, we saw Vibrio abundances increase in the fish microbiota. And the hypothesis here is that what we're seeing are actually increased proliferation of commensal Vibrios in fish, because we did get numerous fish-associated Vibrio strains that were inhibited by the probiotic, including opportunistic pathogens. So those hypotheses are still under examination. All right, so kind of moving on to some stuff that's brand new and super exciting. Just this last summer, I started a project with the goal of growing what we call unculturable bacteria in the laboratory. So what this means, it's estimated that less than 1% of bacterial species that exist in the environment can be actually grown in the laboratory. The reasoning for that, because the nutrients that are available in the environment are vast, and there's cell-to-cell communication in the bacteria, that it can make it very difficult to mimic the appropriate natural environment for those bacteria to grow in the lab. But it's important to get cultures of these microbes that we can work with, because there's a lot of potential for discovery of pre and probiotics, antibiotic compounds, algicides to control harmful algal blooms, bioremediation compounds such as crude oil degradation. The natural bacteria are a great place to discover new compounds. So for this project, I had a graduate student working with me, and we compared growth on standard bacterial plates, more specifically marine agar, to these diffusion chambers, which you can see on the right. Basically, these are diluted media that have very little nutrients in them, and you pour the media into a stainless steel washer, essentially, and each side is covered in a very fine pore membrane. So the pore size was 0.03 microns. That prevents the exchange of microbes, but it allows for the exchange of nutrients in the water. So those diffusion chambers were kind of supported there in the water column. The water in this tank was taken directly from water coming out of our common snook tanks. And the diffusion chambers themselves were inoculated with snook skin mucus. Those were allowed to incubate for 2 weeks. Then the diffusion chambers were taken apart. Part of them were plated to grow the microbes that were there, and part of them were used to supplement new diffusion chambers. And we did that 3 different times during the course of the summer. So this is what the results looked like on the phylum level. These are the number of species. The first column is the number of species that we got just from plating the skin mucus on marine agar. And then you see the number of species we got with each generation of the diffusion chamber, followed by the sum of all the generations and the total number of species collected, and finally the total number of isolates. So 118 total isolates were collected during this process, representing 5 bacterial phyla and 36 bacterial species. Interestingly, 67% of all bacterial species were only isolated from the diffusion chambers, which suggests that the chambers do select for different microbes than what you would be seeing if you just plated things directly on marine agar. So the goal here, of course, was to discover new bacterial species. So in looking at genetic similarity, we were able to isolate 7 bacterial strains with low genetic similarity to described species. So they had less than 95% 16S similarity to described isolates. They were all related to the genus Neptunomonas. And we had at least 2 morphologically distinct isolates within those 7 strains. As you can see here, one was kind of yellow and some of them were more whitish. The reason that this was exciting was because the Neptunomonas group has been identified to produce compounds that may be of human interest. So Neptunomonas naphthoborans can degrade polycyclic aromatic hydrocarbons. One strain of Neptunomonas is algacidal, and one has been identified as a bivalve probiotic. So looking for these new microbes takes me into the next and final project, which is bioprospecting for novel compounds. And at the moment, I'm actually bioprospecting for 2 things. One are novel antibiotics, and 2 are algicides. So from the antibiotic standpoint, I'm working on screening my collection of marine bacteria for antibiotic production against antibiotic-resistant human pathogens, more specifically methicillin-resistant Staphylococcus aureus or MRSA. But this same procedure can be used to screen for antibiotics against any microbe of interest. I currently have about 918 isolates in my collection. I'm always adding more. And the process for this is very similar to what we did in the probiotic and Vibrio antagonism assays, where the environmental bacteria are spot plated and a soft agar overlay of MRSA is poured over top. And here you can see some of the isolates in the collection that have distinct zones of inhibition against MRSA. All in all, so far, I've identified 33 isolates that inhibit MRSA, and you can see they come from a variety of places. Most of them do come from snook, but that's also because I have the most isolates from snook at the moment. They spread through a variety of different genera, including Vibrio, Pseudomonas, Pseudoalteromonas, Bacillus exiguobacterium, and Chidococcus. And so at the moment, I'm in various stages of determining the compounds that are produced. The first step is to determine whether it's a protein or not. And then from there, you can go through various organic chemistry processes to identify if you have a novel antibiotic or if it's already been identified. And I mentioned that I'm also bioprospecting for algicides. This is a brand new project where the marine bacteria that I have and that I'm collecting are being screened for algicide production against the Florida red tide dinoflagellate Karenia brevis. And you can see them there on the right swimming around. That's at a 10x magnification. The reason we're interested in this is because these algicidal compounds are naturally produced in the environment where Karenia brevis lives. There's the potential for harnessing that power to use as mitigation for these harmful algal blooms, which you may be familiar with cause thousands upon thousands of dead fish to wash up, and it's very hard on the economy of Florida during those times. And we do actually have one now that's occurring just to the south of where I am. So what you see here on the left is brand new data that those day 2 counts were taken yesterday. So very new project, but I'm hopeful that through screening these microbes, we'll be able to find some that are algacidal, and then we can work on identifying the compounds they are producing. So in conclusion, I talked about dysbiosis and how it can be associated with disease. The goals of my lab, again, being to help decipher those interactions. What causes dysbiosis? We know it's caused by numerous stressors, but what stressors cause them? What effect does that have on the fish or aquatic animal health in general? Compounds that are produced by bacteria can help to restore a healthy microbiota and improve aquatic animal health. And that was demonstrated through the Probiotics study. Finally, marine bacteria are an underexplored source of novel beneficial compounds for aquaculture through the use of pre and probiotics. Also for medicine, they can produce antibiotic compounds and mitigation, including production of algicides to mitigate harmful algal blooms, and also for bioremediation purposes, identifying xenobiotic degrading microbes. So to finish up, thank you again for having me for your seminar. I just want to do a quick shout out to the funding that I've had over the years, and of course, my collaborators and colleagues, especially the many interns that I've had come through my program that have been absolutely vital to moving this research forward. So that is all. And if I have time, I'm happy to take some questions.
Thank you, Andrea. Appreciate it. So I think we've done this a couple different ways. People can certainly type in questions if they want, but if anybody just wants to unmute themselves and ask Andrea a question, please go ahead and do that. Give people a second to unmute while they do. Oh, I think we got one. No, maybe that was you, Andrea. Andrea, I had a question. So with the oil study, were your data at the microbiota data that you had, was that at the end of the 28 days?
Oh, yes, it was at the very end of the trial.
And so what's your sense of, do you sample along the way? Do you see, like, it must be very dynamic. And so how do you choose? I mean, obviously you have to be somewhat arbitrary at some point, but how different would the results have been sort of partway through? And what did you learn from that dynamic?
That's a good question. And in fact, we do have samples from throughout, but as we were limited by funding, so we sequenced the 28-day samples. And then I also have sequencing data from the seawater control and the chronic CEWAF from 14 days, which would be 7 days after the challenge if they received one. And the patterns are similar in that the CEWAF, even after 14 days, is definitely causing a dysbiotic microbiota. I just haven't analyzed those in as great of detail at this point. So you can learn a lot because the microbiota can shift very quickly. And it is definitely better to try and get that full scale of how things are changing. And so that's something that I'd like to do if given the opportunity and able to find money.
All right, I know Dr. Allison Robertson has a question.
All right.
Hi, Andrea. Great talk. We're lucky to have you give this presentation. So welcome and good job. My question, I'm very interested in harmful algal blooms. So my question will be related to that.
Okay.
I'm wondering, as you consider and are looking for algacidal compounds or activities from these bacteria, are you just looking at a reduction in growth of the Corenia cells, for instance, or are you going to also look at other metrics to see if you're having some effect on those cells beyond just, a reduction in growth?
Yeah, great question. So currently for the screening process, I'm primarily looking at the change in the concentration of Corenia cells over time. as I find something that looks promising, we're going to dive deeper into what's going on. So I have some partners that are going to look at metabolomics of the Corynea cells to determine the impacts that the algacidal compounds are having on the cell physiology. And we're also going to look at brevetoxin abundance, because when you break the Corynea cell open, you're essentially releasing the toxin, and that's not the best strategy necessarily for mitigation. So we do have plans also to monitor the brevetoxin concentration.
Yeah, awesome. Thanks for that. Yeah, I was thinking, like, if you kill these Corynea cells, you're going to spill the contents, and all those toxins will now be in the dissolved phase, you know. But
Right.
another thing, which I'm sure you've thought of, but would be, you know, maybe these compounds or these microbes have the ability to suppress toxin production altogether, and that would be great, even if they don't affect growth. But if you design your study to use growth as your first measure, you may miss the fact that they would actually have a potential to reduce toxin production. Just something that I think is
that's a very good point. And I have talked to the phytoplankton experts about that too. So that is actually something we're looking to add. This first year of funding that we have was more centered towards looking for something that kills the algae, but we do have plans to continue into that research line and see if we can find a microbe that is inhibiting brevetoxin production. So I'm glad you brought that up. That's a good point.
Yeah, awesome. Super interesting. Thanks.
Thank you.
Thanks, Allison. Anybody else have any other questions? Thank you, Andrea. Appreciate it. Wonderful talk. And appreciate you attending a seminar. I wish we could have you here in person, and we'll see if we can do that in the future.
I miss Dolphin Island.
Yeah. All right. Thank you all. Thank you for attending.
Thanks, everyone.